Hello, my name is Andrew Ribeiro. Thank you so much for inviting me. I'm from the Laboratory of Biosystem Dynamics of Tampere University in Finland, and I'm here to present the work that we have been conducting on the dynamics of tandem promoters of E. coli, what controls it, and how can we then use them in synthetic circuits. The work is mostly conducted by Chauhan and Mohammed Bahrudeen, measurements and model specifically. And it's a pleasure to be here. Thank you so much. So closely spaced promoters are abundant in all life forms, including in tandem formation. So they can organize themselves when they're closely united, convergent, divergent, or tandem. And here we study the tandem ones, which are the least studied. But they are capable of interference. So when the RNA polymerase goes and transcribes, it has to stop at the transcription start site, and the closed and open complex formation occur. These are long-lasting processes, and while this is happening, other RNA polymerases cannot go through that part of the DNA. So if they're very close to each other, the tandem promoters, there will be interference due to occlusion in when one is occupied by the RNA polymerase, the other one cannot be. Similarly, even if they're more far apart, when the downstream promoter is occupied by RNA-P, the upstream promoter cannot be producing RNAs, as shown in picture. And so there is that form of interference as well. And we have here, we know that we have 800 genes controlled by 2 or more promoters of tandem formation in E. coli, and we focus on the 102 whose activity is not disturbed by overlapping neighboring genes. So this is the workflow of our study. We started by identifying all genes controlled by tandem promoters using regular DB information. And then we used the YFP strain library to measure their single cell protein levels by flow cytometry. We conducted the measurements both in rich and poor media, which differ in a number of RNA polymerases available in the cell. We also performed RNA-seq to support our studies. And then using the single cell protein fluorescence data, we went on to produce a model by best fitting. And we then went to validate the model with independent data. And what we're currently doing, which I will be explaining at the end, is using the model to construct synthetic circuits, synthetic genes, with the desired dynamics using the tandem formation. So I'll start by presenting the model, the reduced model. We have promoters. The promoters can then be occupied by RNA polymerases, as you see in the top left. And once occupied, it can either proceed to express transcription, to produce transcription, the RNA, or it can be blocked by occlusion, and then the RNA polymerase unbinds. When it starts elongating, there can still be, like I said, interference. And so the RNA-P elongating can go back and make the promoter free once again and not produce an RNA, or it can proceed until the end and produce an RNA. RNAs can then produce proteins, and they can degrade or dilute by the cell division, and the same happens to the protein. So we have the phenomenon of occlusion or elongation, transcription, fall off due to occupation of the downstream promoter, translation, degradation of both RNA and protein. So having the model, we went then to get the empirical data. By flow cytometry, you can see here exactly the data produced. We have 30 genes controlled by tandem promoters that we can measure from the YFP strain library. As you can see, they have very diverse dynamics in optimal growth conditions. Some are very strong, almost bimodal, and others are very weak, almost inactive during these conditions. And then we also measured them in poor media, and you can see that in general, they have a similar dynamics, but consistent with weaker concentration of RNA polymerases, so they have a smaller mean expression level. We try to validate the data using the microscope. So once we had the data, we plotted the gene expression levels as a function of the distance between the 2 promoters. And we measured the mean expression level and the CD squared in the rich and poor media. And we fitted the models to that mean expression level as a function of the distance in the rich media. And we tried to fit exponential models and zero-order models. And what ended up happening is that the best fitting is with a step function. What that tells us is that the main regulatory mechanism of these tandem promoters is occlusion. So if it's small enough to occlude, the expression is very weak. if it's long enough to not exist or distant enough to not occur occlusion, then the expression can be stronger. So that form of interference is the strongest regulation. And also, of course, it matters a lot, the strength of the promoters. Having the model suggesting that that interference can be tuned by DTSS, the distance between the promoters, and by the occupancy, we then went to what we're currently doing now is validating these results. For that, we're using synthetic constructs in single copy plasmids. We're using the standard promoters that are used in molecular biology in E. coli, which is the LEC and the TET and the BAD promoter. And we're assembling them using for measuring expression mCherry. So they're currently assembled, and we're going to start very, very soon measuring their dynamics. And as you can see, they differ in the two main variables that we're describing from the model. Distance between the promoters and strength of the promoters that we can control with the induction strengths by external regulation. And that's how we're going to be testing the model. And once this is validated, that's where we're going to go and do the most important stuff. So the most challenging thing in, for example, for advances in synthetic biology is that it is still very, very hard to predict gene expression from the promoter sequence. It's extremely challenging. And so most circuits are so far limited to the few promoters like we were seeing, the LEC and the TET and so on. But in our case, it appears that we can tune the kinetics of the tandem promoters by the nucleotide distance between them and by the kinetics of the two component promoters. But we don't know that kinetics of the two component promoters unless, and this is the key thing, we use an existing strain library whose expression has already been measured. And that's exactly what we're proposing here. There is already known expression levels of these genes, but it's limited to the existing ones. So what we're proposing is that combining this library with our model, we can engineer tandem promoters with a predictable dynamics, and thereby we can do the dynamics that we want in both mean and noise, combining the strain library with our model. And so we believe that this is a very promising method to create synthetic genes with a wide range of predictable dynamics that will then be able to allow us to construct very diverse synthetic circuits. So we believe that they can become, combining these genes with the model in tandem information, this can become very interesting set of building blocks of genetic circuits with a predictable dynamics. Thank you very much for your time.